So what I have up on the board is the basic idea of what we're doing in the transformation lab. You're going to have two microfuge tubes that actually look something like this. Okay, so that's actually a little microfuge tube. So you're going to have two microfuge tubes. One is going to be labeled positive for positive DNA, and one is labeled negative for negative DNA. Throughout the procedure, we're going to add E. coli to both tubes. We'll add calcium chloride, which they call in the lab manual, they call it transforming solution, but it's actually just calcium chloride. We're going to add something called LB broth to both tubes. And then we're going to treat both tubes equally by heating it up and cooling it down. So we're adding all of these things to both tubes equally. The one thing that we're doing differently to our two tubes is adding plasmid to one tube. Notice we're adding the plasmid to the positive tube. And from lecture you should know, remember that a plasmid is an extracircular loop of DNA for bacteria, and it can hold all kinds of information like antibiotic resistance, mating factor, certain accessory structures and things like that. So we'll be using a particular plasmid that's carrying certain genes like the GFP gene and antibiotic resistance, and we'll talk about those a little bit more in class. That's also explained in your lab manual. So that's the basic idea. We've got our two microfuge tubes. We're treating them the same with each of these four factors, but we're adding plasmid to only one tube, the positive tube. Once we've gone through this procedure, we're then going to have 4 plates. There's going to be 4 different plates that you're going to be spreading bacteria on. One plate will be labeled LB amp positive. Another one is LB amp negative. Another plate is LB amp ERA, and ERA stands for arabinose positive. And the other one is simply LB negative. Don't worry if you didn't get to copy all this stuff down, because it's found in your lab manual. So you've got very detailed, clear instructions. But make sure you go through it so you understand what we're doing in each step before you get here. So let's go ahead and go back to the lab bench, and I'll show you some of the things we're working with. So what we've got here are some of the things that we're going to be working with in lab, including a micropipetter. For the micropipetter, well, sorry, first we have our little microfuge tube. So you'll have 2 of these microfuge tubes. One will be labeled positive, the other one will be labeled negative. This is the actual rack that they fit into, so we're going to be using our microfuge tube racks. We'll be using a micropipetter. If you've never used a micropipetter before, it's okay. You've got a plunger up on top, and what that does is when you want to actually suck something up, first you need to get the pipette tip. Right now there is no pipette tip on the bottom. So this is a box of sterile micropipette tips. You open up the top, and I'm gonna just press the tip onto my microfuge, onto my micropipetter. To actually suck something up, if I wanted to draw up some liquid, when you get into class you'll feel the plunger. The plunger actually has 2 stops. It has the first stop, and for the first stop is to suck something up. So I would take my micropipetter, put it into whatever liquid. Let's say if I wanted to use some liquid. So I would put my micropipetter into whatever liquid I wanted to suck up. I press down the plunger to the first stop, put it into liquid, release the plunger, and I now have a small measured amount of liquid in my pipette tube. So now if I want to expel this, if I want to get it out of my micropipetter, let's say I'm going to put it over here, I would actually go ahead and push the plunger all the way down, and it dispenses the liquid into whatever you're going to put it into. So if I want to suck something up, I press the plunger down to the first stop, put it into my liquid, let it go. I have a measured amount of liquid. To dispense it, I'm going to press the plunger all the way down, and all my liquid is out of my pipette tube. So now, since I've used this micropipette tip already, it's now contaminated and I want to get rid of it. So we would get rid of it into a biohazard bag that unfortunately I don't have here. So to take off the pipette tip, there's a little button up here that you would use, and just to kind of show you, the tip will just get pushed off. Okay, so the micropipette tip should go into a biohazard bag that you'll have on your table. So that's the micropipetter. Practice a little bit when you get into class. The other thing we want to show you is how to use an alcohol burner using ethyl alcohol as well with the spreader tool. The one thing to be very clear about is safety. This is an alcohol burner, and there's alcohol in this burner, so that's actually what lights, what actually holds our fire. We want to keep our two sources of alcohol separate from one another. We're going to have flame here and a jar of alcohol. Very important to keep them separate. So we'll light our alcohol burner. Okay. So you see we've got a flame on top of the alcohol burner. I'm going to take my spreader tool, which is metal. I dip it into alcohol. Okay. Let the excess drip off, and I simply pass it through the flame. Okay. You might be able to see that it's actually burning blue, so we're burning off the alcohol. Just let it burn off. This is now hot. We do not want to put a hot spreader into this jar of alcohol, or else we could literally have an explosion. So we want to be very careful about safety procedure. So again, if I want to sterilize my spreader tool, I'm going to dip it into alcohol, let the excess drip off, pass it through the flame. Don't hold it in the flame, just pass it through. Let it burn off, and so we let it cool a little bit. To spread bacteria onto our plate, we're going to use this spreader tool. And so our plate would actually be upside right with the lid on top. Open it up, and you'll have some liquid on your plate. You take your spreader tool, and you simply skate it across the surface while you turn your plate. So you should have some liquid on your plate. You're going to be spreading it out, spreading it out, spreading it out. And so once you actually get it all over the plate, and you've spread it out across the auger, we can close that up, and we need to sterilize this again. So we dip it into our alcohol, pass it through the flame again, we let the alcohol burn off, and now your spreader tool is sterile. So just a couple of things to keep in mind. Remember your safety. We want to keep the alcohol separate from our flame. We can close up our alcohol when we're not using it. And then to turn out your alcohol burner, it's just a big birthday candle, so you just blow it out. Okay. So those are some of the things that we're going to be using in our transformation lab this week. There's plenty more explanation. Make sure you read through your lab and come into class kind of having an idea of what we're going to be doing in lab. Okay, thanks.